Hello and welcome to this webinar, which is based on a presentation held at the Society of Toxicology conference in San Antonio earlier this year. My name is Andy Forreryd and I'm a scientist working in the research and development team of SenzaGen. So the aim of this presentation is to illustrate a proof of concept on how genomic technologies can be applied to build versatile but also highly accurate in vitro testing platforms. So we actually believe that this has a potential, this strategy, to actually transform the way toxicity testing is performed today. So throughout this webinar, the leading example for this will be our SenzaGen's main product, Genomic Allergen Rapid Detection. So before I go into any specific details of this testing strategy, I would like to give you a short introduction to the company. So at SenzaGen, we develop in vitro assays for various immunotoxicological endpoints using genomic technologies. We are based in Lund, Sweden, and we were formed 2010 as a spin-out from Lund University. So SenzaGen currently employs 20 people. And our lead product, which I'm here to talk about today, is Genomic Allergen Rapid Detection, which is a state-of-the-art testing platform for assessment of chemical sensitizers. So, working within the field of predictive toxicology, the main challenge for us is to translate a complex biological endpoint into a tool that can be used for risk assessment. So ideally, we want our assays to provide biologically relevant results with a high predictive performance. We also want them to be simple to perform, and for various reasons today, it's of course ideal if they do not involve animal experimentation. So what it then often comes down to is a trade-off behind the simplicity of the assay and the informational content it generates, on which we can then base our final decision. So while animal models traditionally position themselves in the lower right corner of this plot, the majority of the in vitro assays available on the market today, they are based on one or perhaps two biomarkers and provide very little mechanistic information. So the problem with using such a simplified in vitro model is that they normally fail to represent the true complexity behind the biological endpoint. So if you instead then start to add parameters to your readout, you can begin to build a view that better represents the underlying complexity. And in the end, of course, you would like to see the full picture. So in terms of skin sensitization, which is the endpoint that we were interested in from the beginning, we do have a rather good understanding of how this full picture actually looks like. And it involves the intricate response by the immune system to a foreign substance. So I try to illustrate this, this response here in this figure. So what actually happens when you're first exposed to a sensitizer is that it will penetrate the skin and bind to endogenous protein to form a hapten protein complex. So this complex is then recognized by dendritic cells, which in response to inflammatory mediators will migrate to the local lymph node where they activate allergen-specific T cells. So this is the initial priming of the immune system. So each time that you're then exposed to the same chemical, it will result in a rapid activation of the allergen-specific T cells, which will result in an inflammation in the skin, which is characterized as allergic contact dermatitis. So for the rest of this presentation, I would like to focus on one of the key players in this process, which is the dendritic cells. So we know that dendritic cells, they typically serve as sentinels where they guard the peripheral tissues from non-self molecules that enter these tissues. So when these cells then encounter this foreign substance, they go through different type of functional but also phenotypical changes that allow them to initiate the downstream immune response that give rise to allergic contact dermatitis. So the question is, of course, then how we best monitor these changes that happens in the dendritic cells. So we know that these cells react in a multitude of different ways when they're exposed to a sensitizer. First of all, they have different type of receptors for recognition of foreign substances. And these receptors may in turn activate various type of self-defense mechanism of the host. You might have different type of cellular stress responses, such as oxidative stress. And they may also express different type of receptors that allow them both to communicate, but also to migrate towards other cells of the immune system. So we know that all of these changes are carried out by protein, and the level of protein expression is, of course, regulated by the transcription of DNA. So based on this, we believe that genomics is a very efficient way of getting a complete view of what's actually going on in the cells when they're exposed to a chemical sensitizer. So with all of this information and this knowledge, we early on hypothesized that there should exist genes in dendritic cells that are differentially regulated depending on if these cells were stimulated with a sensitizer or a non-sensitizer. And of course, if we were able to identify these genes, they could also be used as a predictive tool. So in our first ambition to actually test this hypothesis, we identified an in vitro model for dendritic cells, which we call Senza cells. So we stimulate these cells with a panel of reference chemicals comprising 20 sensitizers and 20 non-sensitizers, and we measured the complete transcriptome of these cells using microarray technology. So for each of these chemical stimulations, we measured 29,000 genes. So this is in contrast to the traditional way of performing toxicity testing, because there you normally know what to look for and you measure only one biomarker. So now instead we measure the complete transcriptome of the cells for the purpose of identifying novel and more predictive biomarkers for skin sensitization. So the first thing we did was to visualize the gene expression data using principal component analysis. So in this figure, we have colored our stimulations according to the sensitizing properties, where a sensitizer is red and the green is the non-sensitizers. So basically what we see in this plot is that each of the dots summarizes the gene expression from one single chemical stimulation. So what we ideally want to do is to separate these two groups, because that means that we have genes that are differentially regulated. So if we use all these 29,000 genes directly from the genomic arrays, you can see that it is impossible to distinguish the sensitizers and the non-sensitizers. But by using advanced tools for data analysis, we were able to identify 200 genes that were differentially regulated depending on if these cells were stimulated with a sensitizer or a non-sensitizer, and we collectively refer to these 200 genes as the GARD prediction signature. So we could also, of course, visualize the same 200 genes in a heat map, which further illustrated differences in expression profile induced by the non-sensitizers and the sensitizers. And what I think is extremely important here is that we use a completely data-driven approach to identify the 200 genes in the predictive signature. But when we look at the identity of the specific genes, we can see that they are indeed functionally relevant to the immune response, and they are involved in exactly the same mechanism as described previously. So with all of this information at hand, we basically set out to develop an assay that utilizes the readout of this biomarker signature as a predictive tool. So to do this, we use something that is called pattern recognition and machine learning. So what we basically do is that we use the information from the GARD prediction signature, as training example. So we train the machine learning algorithm on the gene expression profiles of sensitizers and non-sensitizers, and the machine learning algorithm learns to discriminate the expression profiles. So when we're then about to classify an unknown testing substance, the gene expression profile induced by this substance is compared to the training example in the machine learning algorithm. So in this specific sample that I tried to illustrate in this figure, the expression profile of the unknown testing substance is most similar to the expression profile of the non-sensitizers in the training data, and that means that this compound is classified as a non-sensitizer. So this technology is also used by other type of companies such as Tesla to design their automated driving cars, Google to tailor your searches, but it's also used within the QSAR community for developing predictive models. So based on this overall strategy on how we actually used these genes as a predictive tool, we have now also a testing strategy on how you can actually guard your products in only 6 steps. So I would like to go a bit deeper into this specific mechanism. So in practice, what we do today is that we stimulate our in vitro model of dendritic cells, our Senza cells, with any chemical substance that are to be tested. So we stimulate the cells with one single chemical stimulation or concentration, which we call the GARD input concentration. So to find this concentration, the first thing that we do is to screen for cytotoxic effect. So we typically aim at a concentration inducing a relative viability of the cells of 90%. So if the compound is non-cytotoxic, we use a default concentration of 500 micromolar. And if this is not possible, we simply use the highest soluble concentration. So when we have identified the GARD input concentration, we perform our cellular stimulations again. So this time we perform a quality control of the cells. So what we do is basically we use flow cytometry to measure common maturity markers of the dendritic cells for the purpose of ensuring that the cells are immature, where they actually are capable of taking up and process antigens. So if the phenotype of the cells looks good, we incubate the cells for 24 hours. So after that, we isolate the RNA from the cells, and we measure gene expression within the GARD prediction signature using either Affymetrix array or the nanostring technologies. So the Affymetrix platform is we use that during biomarker discovery processes where we measure the complete transcriptome of the genes. But when we perform routine analysis, we instead use the nanostring platform, which enable a more tailor-made readout of only the genes in the predictive signature. So in the next step of this procedure, we basically compare the gene expression of the unknown sample to the gene expression profile of the samples in the reference dataset. And this is what I was talking about before. We use a pattern recognition mechanism that is called support vector machines. So what I try to illustrate in this plot is how the support vector machines actually work. So what it basically does is to find a hyperplane that best separate the sensitizers and the non-sensitizers in our training data. So the classification of the unknown samples are then based on the distance to this separating hyperplane. So this means that a positive value is classified as a sensitizer, a negative value is classified by the algorithm as a non-sensitizer. So For final classification then of a specific sample, that is based on majority voting from triplicate stimulation for each chemical. So what we see in this figure is basically data from a recent in-house validation project. So if we look at the plot, what we see to the left of the dotted line, they are the true sensitizers, and to the right is the non-sensitizers. So the ones that are has a positive GARD decision value, they are the one that are classified by the algorithm as a sensitizer. And the one that has negative GARD decision value is the ones that are classified by the algorithm as the non-sensitizer. So what we can see in this figure is that only one specific compound were falsely classified as a negative. So what we have done today to facilitate for the end user is that we have now also implemented our prediction model into the cloud. So what the customers basically only have to do is to upload upload the gene expression data from the non-string and into our application, and the application which hosts the prediction model will perform and provide you with the classifications. So does this strategy works? Yes, we have tried it in many different data sets today. So this information is all published in various type of scientific peer-reviewed journals. And today we have assayed a total of 127 compounds with an accumulated predictive performance of 86%. So it's of course relevant to compare this predictive performance to the gold standard in vivo model that are available today, which is the local lymph node assay, which is only has a predictive performance corresponding to 72%. So it's of course also important and also interesting to compare how the GARD assay performs in comparison to other in vitro assays on the market today. So this is something that has recently been done by an independent scientist, David Roberts from John Moore University in Liverpool. So I'm sorry. So what he basically did was that he identified the overlapping substances that had been tested in both the GARD assay, but also in the three independent or in the three OECD guideline approaches. So his overall conclusion from this exercise was that GARD outperforms each of the three non-animal OECD guideline approaches, either if they are used individually, but also when they are combined into an integrated testing strategy based on majority voting from a 2 out of 3 integrated testing strategy. So this essentially means that using the GARD assay is more predictive than using a combination of the three currently recommended non-animal OECD guideline approaches. So based on this data, we have also now initiated a formal validation procedure that is based on OECD guidance documents. So the first step in this procedure was basically to recruit 2 external naive laboratories. So we established a cooperation with Burleson Research Technologies in the US and also to Eurofins in Germany. So what we did was that we went to these laboratories and we set up the GARD assay and we trained the personnel on how to perform the assay. So the next step in this procedure, we asked these laboratories to classify a total of 11 compounds of unknown substances, and of these compounds, 8 compounds were sensitizers and 3 compounds were non-sensitizers, and we asked them to do this classification in 3 consecutive laboratories. So what we see in this figure, once again, I would like to remind you how to interpret this data. So a positive value in the GARD assay means that the assay classified the compound as a sensitizer. A negative value means that it's classified as a non-sensitizer. So based on this, we can actually see in this figure that all the compounds were correctly classified as sensitizer and non-sensitizers in all of the 3 consecutive experiments at both Burleson and the Eurofins laboratory. So this essentially means that the assay was quite easy to transfer to these laboratories. So based on this result, we initiated the second phase of this validation procedure, where the validation management group selected a total of 28 compounds that were to be tested in each of the 3 laboratories. So this is basically a ring trial where all of the compounds should be classified in 3 consecutive experiments in a complete blinded fashion. So after we have performed all of these testing, the results were submitted to the statistician, and the validation management basically enabled the decoding of the substances. So what we see in this figure is the result from the classification of these 28 compounds. So once again, we can see that the majority of the sensitizers and also the majority of the non-sensitizers were accurately classified at each of the 3 laboratories, but also in the 3 different experiments. So we can also see, if we compare the stimulations, or if we look at the specific chemical stimulations, we can see that the standard deviation between the experiment, but also between the different laboratories, were really small, which indicate a high reproducibility both between and within laboratories. So the same data could of course also be illustrated in a table. So what we basically done in this figure, or this table, is that we coded the samples. So for each chemical, we can see that the green color corresponds to that 3 of the 3 experiments were correctly classified at a specific laboratory. So we can see that the majority of the colors in this, or the most highly represented color, were actually the green ones, which means that the majority of the compounds were correctly classified in all of the 3 experiments in all laboratories. So if we then look at the within laboratory reproducibility, we can see that it spans between 82 and 89%, and the between laboratory reproducibility between the 3 laboratories were 92%. So we of course also calculated the cumulative predictive performance of the assay, which reached an impressive 94%. We can also see that it's a very good balance between the sensitivity and the specificity. So based on these, the overall conclusion from the validation of GARD is that the assay is easy to transfer to naive laboratory. It's highly reproducible both between and within laboratories, and it's also highly predictive for identifying skin sensitizing chemicals. So following this validation of our GARD skin assay, we have also worked really hard to be able to provide also information of relative sensitizing potency. So as it is now, GARD skin is only capable of providing a yes or no answer if the compound is a sensitizer or not. So what we basically have done now is that we have identified a predictive biomarker signature that are capable of subcategorizing the sensitizers according to the CLP regulation for labeling of chemicals within the European Union. So it's capable of discriminating between moderate and weak and strong sensitizers. So the intended use of this prediction signature is within a thyroid approach. So what you basically do is that you use you use the GARD skin assay for identification of the sensitizers, and then you use the GARD potency signature for further subcategorizing your sensitizers into strong and weak sensitizer. So we have, of course, performed some initial cross-validation of these assay, and the accuracy this far is expected to be somewhere between 75 and 80%. So this GARD potency signature is now also included in the second phase of the formal validation procedure. So what we do now is basically that the first 28 compound that were used during validation of GARD skin is now further subcategorized into potency groups, and we also classified an additional of 30 blinded substances into these two categories. So what we basically have done now is that we have finished all the laboratory work related to the potency signature, and we are currently summarizing all the results, and we expect to submit the full validation report on both GARD skin and GARD potency at the early April. So we will submit this to ECVAM for scientific review, and we expect to have an OECD test guideline approval ready already 2019. So following these results of these, so what I've shown you this far is more or less finished applications that are available for use and are just awaiting formal regulatory acceptance. What I would like to show you now is basically the assays or the add-ons that we actually have in our research and development pipeline. So the first application that I would like to show you today now is what we call different modification to the GARD skin protocols, which would allow us to assay different type of more complex samples, such as, for example, medical devices, but also different type of complex mixtures. So when we look at medical devices, we are currently working with implementation and optimization of different type of extraction protocols for solid material that is based on international agreed standards. So this far we have tested a different type of absorption chromatography columns for blood purification processes, and also different type of silicone-based material. So what we essentially do is that we perform an extraction of these medical devices, and then we test the extract in the GARD skin assay. So this add-on is available today, and we discuss this type of project on a case-to-case basis. So for complex mixtures, we of course also have an protocol that allow us to assess not only pure substances, but also different type of mixtures. So this strategy is based on custom design extraction steps that is based on the nature of the sample. So GARD is so in GARD we can use a lot of different type of solvents, and we have tried. So the normal ones is DMSO and water, but we have also tried, for example, DMF, glycerol, ethanol, and sesame oil. And these we have initiated and completed a project that involve different type of polymerized gels, but also oils from the cosmetic and food industry. And these are very complex samples that are practically insoluble in standard solvents. So this protocol is also available for use today, and we discuss the project on a case-to-case basis. So by that, I would also like to show you an example from a recent project that we performed together with a customer. So the customer provided us with a UVCB sample to test in the GARD assay. So what we did first was we tried to dissolve this compound in the MSO, but the compound was practically insoluble, and as we can see, it didn't induce any type of cytotoxicity in the GARD input finder. We tried to classify this compound as well, and it was falsely classified as a negative substance. So we tried the same using water as solvent, and we could observe pretty much the same here. The compound was insoluble, it didn't induce any cytotoxicity, and it was giving only a very weak answer in the GARD prediction assay. So instead we went back to our customer and we asked them to provide us with some other details of the compound. And indeed, when we actually went back and considered the physiochemical properties of the substance, we could find a solvent that were more suitable for this specific compound. So we used DMF, a mixture of DMF and glycerol, and we could see that we could finally get this mixture into solution. And as you can see, it became bioavailable to the cells. It starts to induce cytotoxicity, and it was then finally correctly classified also in the assay as a positive. So this is basically how we work around this type of complex samples. So in addition to this complex samples, we are also working to define the applicability domain of the GARD assay for pure chemical substances. So what we have shown this far, based on all the substances that we have tested, is that our assay, GARD skin, is applicable across all chemical reactivity domains, and it's also able to correctly predict pre and pro haptens. So in addition to this, we have also initiated different type of customer product where we have a collaboration with LASA to identify different type of data gaps in this chemical space, where we would like to demonstrate the usefulness of GARD on even a larger type of chemicals. So in addition to this, we have also worked with implementation of the conformal prediction framework. So this framework instantly provides you with a warning if you try to predict a compound that is outside the applicability domain of the assay. So this will be really useful for customers, because then we can directly see and tell the customer if a specific compound is outside of the domain that would result in a certain prediction. So for the next two applications that I would like to show you here today, which is the GARD air and the GARD protein application. So these two applications are essentially based on exactly the same standard operating protocols as the GARD skin assay, but the only thing that is the training data and the biomarker signature, which we tailor to the specific biological endpoint. So starting with GARD air, we investigated the possibility to extend the applicability domain of the assay to classify respiratory sensitizing chemicals, but also respiratory irritants. So based on this endpoint, we actually believe that we already had a really good cellular model in the GARD assay that are capable of identifying also respiratory sensitizing chemicals. So because the dendritic cells that forms the core of the assay, they are responsible for the polarization of the T cell response into either a T helper 1 response or a T helper 2 response, which are the two polarizations that are responsible for either skin sensitization or respiratory sensitization. So for respiratory sensitization, we used a similar approach as when we performed the GARD skin assay. So we identified a panel of reference chemicals which we stimulated our cells with, and we were able to identify a predictive biomarker signature that were capable of discriminating between respiratory sensitizers and non-respiratory sensitizers. So in the next step, we of course try to validate this signature. So we classified an external test set, and we could estimate the predicted performance of this assay to 84%. And what I think is really important here is that the majority of all our skin sensitizers that we have tried to classify in this assay, they have actually classified as negative. So this means that this assay is not only capable of identifying respiratory sensitizing chemicals, it is also capable of discriminating between respiratory sensitizers and skin sensitizers, which is something that no other assay available on the market are capable of doing today. So based on these results, as being the first predictive in vitro assay for identification of respiratory sensitizers, we recently received external funding from the Horizon 2020 project in order to further work with this assay. So what we use this funding for is basically that we would like to push the respiratory sensitizing signature into more a practical application, and further push this through the validation procedure. But we're also working with actually identification of genes that could be used also for identification of respiratory irritants. But be able to assay these dual endpoint would be a really great opportunity to perform a more complete risk assessment of this type of chemicals. So for respiratory irritation, we initiated a collaborative study with AstraZeneca. So they provided us with some well-known respiratory irritants, which they have really good clinical data on in their library. And by using, again, similar strategy, we could identify a predictive biomarker signature that were capable of discriminating between the irritants and the non-irritants. So these two assays are actually also kind of close to getting to the market, and we have performed some initial projects together with external customers. So in the figure to the left, I would like to show what we did with respiratory sensitizers. And so what we did was that the customer sent us 5 test substances that we predicted blindly in the assay. And together with these 5 test substances, we also predicted a total of 10 chemicals that we know their ability to induce sensitization. So of these compounds, a total of 8 were correctly predicted as according to their sensitizing properties, and for the test substances, all were classified as negative, which seems to fit quite well into the data that the customer had available also. So in terms of the respiratory irritation, another customer provided us with 3 substances, and we predicted these in the respiratory irritation assay together with a negative and a positive control. And all of these test substances were predicted as positive, which again seemed to fit quite well into the data that the customer had available. So following this, I would like to introduce also the last project that we have now in our research and development pipeline, which is a GARD protein assay, which is basically a R&D project that aims towards developing a predictive assay for assessment of allergenic proteins. So once again, we established a collaboration with the industry, and we received 8 industrial proteins where we investigated the transcriptional profile of. So based on these, we could identify also a set of differentially regulated genes in our dendritic cell line, dependent on if the cells were stimulated with a protein allergen or a non-allergen. So here I think it's important to mention that the training data set we use here was really small. It was eight positive allergens. But when we look at the data, the statistics look really good. So there's a big difference in the expression profiles. So if we have also now tried to initially challenge this data or this prediction signature with an external test set. So this basically was what we did, that we used two protein homologs. One from so we used two trypomycins. So one was from shrimp, which is a known allergen, and one was from trypomycin, which from pork, which is a non-allergen. And as you can see, the shrimp trypomycin was correctly classified as a sensitizer, and the pork trypomycin was correctly classified as a non-sensitizer, which kind of highlights the possibility of this signature to discriminate closely related protein homologs. So by that, I would just like to summarize the main take-home messages from this presentation. So what I hope that I've shown you is that genomic biomarker signature enables a readout with really high information, and that this high information will be reflected in a consistently high predictive accuracy. And this far, GARD has the highest reported predicted performance for skin sensitization, and it also provides a quantitative assessment of the relative sensitizing potency. And I hope that I've shown you through our research and development pipeline that using genomics it makes the assay highly versatile, and that it will allow in the end for a broad applicability domain. And I would like to finish off this presentation by stating that this technology is readily available today. Don't be afraid to use it. And by that, I would just like to acknowledge my co-workers at SenzaGen and our collaborators at Lund University and our validation manager, Irving Roggen. And of course, I would like to thank you all for listening, and I would be happy to take any questions.